Welcome to today's session, children. Today, the first two topics we are going to study is about antibiotics and vaccination. Antibiotics are biochemical substances released by microorganisms in very small quantity, which kill or stop the growth of other microorganisms. This was discovered by Alexander Fleming in the year 1929 very accidentally. He was growing a culture of Staphylococcus aureus and suddenly he saw it being contaminated by a fungus. He slowly teased out this fungus and observed it under the microscope and found it to be a fungus called as Penicillium notatum. Later on, he understood that this fungus was producing some chemical substance which was preventing other organisms from growing in it, like bacteria or any other bioorganism. This was the discovery of the wonder drug penicillin, and it was used to treat thousands and thousands of people wounded in the World War. So hence was discovered penicillin and it was appreciated all over the world. Similarly, other examples of antibiotics are streptomycin, tetracycline, erythromycin, etc. The concept of vaccination is the process of inoculation of dead or weakened microbe into the body of human to generate antibodies to fight against microbes. The antibodies are generated by our WBC cells and they create a system to fight the new organism that has entered into our body. The memory cells of our defense system remember this foreign organism and protect our body from further attack by the same organism. Vaccination are given against diseases like polio, tuberculosis, hepatitis, etc. Polio is a disease which has been eradicated. It is not anymore existing in the world around us. Government has taken many steps to see that the disease is eradicated by repeated vaccination of children. Smallpox was one such disease which was eradicated previously. Let us go on to the next topic, food poisoning. The presence of microorganisms or their toxins in food leads to very serious illness and even death. This condition is called as food poisoning. Food poisoning occurs with vomiting, diarrhea, and high fever. So, if you consume adult a food which is containing microorganism or its poison, which can lead to conditions mentioned above, the process is called as food poisoning. It is important that we preserve food to prevent it from spoiling. Avoid consumption of food from outside sources to prevent food poisoning. If symptoms are noticed, fluid substitutes to be provided to patient and immediate hospitalization advised. What are some of the food preservation techniques? Food cultivated in surplus has to be preserved for future use. Different types of food materials require various types of preservation techniques for enabling their availability throughout the year. What are some of the methods of food preservation? We have salting and sugaring. High concentration of salt or sugar kills microbes by causing their cells to burst. Example, mangoes, amla, dry fish, jam, jellies. Chemical preservatives are used in fruit drinks, squashes, pickles. Some examples of such chemical preservatives are sodium benzoate, sodium metabisulfate or vinegar, which prevents the growth of microorganisms. Heating kills microorganisms and preserves food. Pasteurization of milk is a method of preserving milk by heating it. Cold treatment like freezers and refrigerators are used to preserve perishable food, example meat, milk products, fruits and vegetables. Drying in sun is an ideal method of preserving foods like pulses, cereals, vegetables like chilies, bitter gourds for longer shelf life. Canning of food is done in many parts of the world. Western countries mostly make use of canned food. Food like fish, meat, milk products, fruits, vegetables are canned and preserved. Let us go to the last topic of the lesson, which is nitrogen cycle. 78% of the atmosphere contains free nitrogen, but nitrogen cannot be used by living forms as they have to be converted into nitrates and nitrites. By living forms, I make it clear that it is plant that makes use of this nitrogen. So it has to be converted into nitrates and nitrites if it is to be used by the plants. Nitrogen cycle is divided into four different parts: nitrogen fixation, ammonification, nitrification, and denitrification. Let us look in detail into the nitrogen cycle. The free nitrogen, which is available as N2, has got different methods of fixing into the green plants. Industrial fixation takes place when nitrogen-rich fertilizers are used in the soil, like urea. These are containing NPK as fertilizers, so plants can avail this nitrogen when it is used as fertilizers in the field. Certain blue-green algae and leguminous plant are able to fix free nitrogen for the plant. Blue-green algae has got a mechanism of fixing nitrogen in their body, and legume plants have got the rhizobium bacteria which fix nitrogen for the plants. Example, legumes. Lightning is a method of fixing nitrogen into the soil. All these methods enable the plants to convert free nitrogen into plant proteins. Plants are eaten by animals and these are fixed as animal proteins in the body of the animals. On death and decay of both plants and animals, these nitrogen or proteins is converted to ammonia. The process is called as ammonification. Ammonia is converted to nitrite in two steps. One is by first converting it into nitrite and then converting into nitrate. These are done by the soil bacteria, and the NO3 that is formed is released into the atmosphere as N2 again by the activity of soil bacteria. So this is a brief of the nitrogen cycle. You can go through the notes in detail for the process of nitrogen fixation, ammonification, and nitrification, denitrification. Thank you, children, for patient listening of the class. Complete all the work that is allotted to you. Good day to you.